Morning, how are you? Good. It's like Mainak and Mayukh are here. Hello? Looks like Susan's here.
Oh, Susan.
Hi. Can you hear me today?
Yes.
Oh, wow. I've had a few logins with my computer tech. I got a new service.
Oh, okay.
It's about $20 a month, and you can have some sleep. Oh, yeah.
Yeah, that's a plan. Let's see. Can you see the pattern on that one?
Just along the edge.
That's a little trouble.
If you've got some tiny ones that are like maybe you could pop one in the mail.
I have some real tiny ones, but
there has much. Oh yeah, that has some patterning on it.
Dark. Yeah, here's some other patterns.
Yeah, those are nice. I do have some shells somewhere. They weren't necessarily collected for their patterns, so who knows what I got.
Yeah, I know most of the ones you collect on the shore. These were collected in shell shops.
Okay. Well, these mine are amateur collectors who are wondering at each.
The general problem is this. Okay? You take a pattern like this. Can we prove that it corresponds to some pattern, one of the Wolfram patterns? Can we show that the time course matches that of some real pattern? Or to put it another way, can you go backwards? Can you see the pattern and figure out the rules?
Oh, okay.
Okay?
Oh, I'm busy writing an essay about integrins and collagen 1 and
Yeah, here's a real tiny one.
so oh yeah.
So I think now the shells that I collected, I deliberately got defective ones. But I couldn't do that much with little ones. With big ones, I've got some, the size of my hand. I have shells that at some point during the life of the snail, the shell broke and it was repaired, and the pattern had to resume. And so you can see the break and you can see the resumption of the pattern. So those are, they're kind of natural experiments. Okay? So
about what you guys are talking, like,
we're talking about,
can I get the context?
the context is patterns on snail and clam shells. Here's some stuff with it. See?
Yeah, I can see that. Yeah.
Okay? I collected shells from shell shops around the world.
Yeah. Actually, I think cells have like very unique patterns in every species, and I think everything is quite organized.
Yes,
So, like,
it's similar to patterns generated by Wolfram's rules. Some of them.
yeah, like somehow, like I see that one species have a pattern like horizontal, like vertical line.
Here's another pattern.
Yeah, that's very nice one. I think it's your hobby.
Well, here's another one with just dots. So the question which is unanswered. People presume these patterns are made by the cellular automata rules of Wolfram, and some of them look similar to them. But the question is, can you image the pattern and then derive the rules from the pattern? In other words, do the inverse problem.
Okay, so like give the input of a cell, and after that you can define the rules from it, or the something like that.
Yeah, well, the question is, okay, here's the pattern. Can we use the pattern to derive the rules that made it? Okay?
Okay.
The ordinary approach to this is you make a bunch of patterns in the computer and you find one that looks similar to it by your eye.
Yeah, more like a lexicon approach or a dictionary approach.
Yeah, so the inverse problem. Talk about the inverse problem. How do you add into the rules?
I think instead of doing this like a can or something like that, if we can generate fake fake patterns from these original patterns, and after that if we can discriminator, it can suppress the generator. So I think that from that we can define some rules.
Well, maybe. No one's tried it.
Yeah.
Yeah, maybe, because I'm familiar with the concept for 2 years or 3 years.
Yeah, see,
So yeah, you are more experienced.
Susan is building microscopes that can take a three-dimensional shape and represent it in the computer. So it should be possible to take these shells and get their three-dimensional shapes. And then one thing you can do, for example, with a shell like this one, if I rotate the shell, you can imagine unrolling the pattern. As if the shell was made of paper and you could unwrap it.
More like unwinding.
Yeah, unwinding. Yeah. So basically, we want to go from the imagings to 3 dimensions to unwinding in 2 dimensions.
Yeah, it's a really interesting problem, I think.
Okay. Can you get shells like that in India?
What?
Can you get shells with patterns in India?
Yeah, definitely I have lot.
Oh, okay. Yeah. Well, you have a camera because I'm looking at you. So.
Actually, this is the webcam of the laptop. It's not too good.
That's not that bad. Right?
Yeah.
So get some shells and think about it.
Yeah.
Definitely.
Yeah, well, that was good. Thank you, Dick. Yeah, that's an old problem for the group. We've been talking about that for a couple of years, where, we want to figure out how to image the whole surface of the shell and, turn it, this is kind of like the thing we have with the ball microscope and the flipping microscope, where you have these images that you're collecting from different perspectives and you're trying to stitch them together.
Yeah, we made a crack at that with Tom with the, what did he call it? Morpho something? Yeah.
Morphozoic? Yeah.
Yeah, with Morphozoic. But we never got to the point where you can take a pattern and deduce the rules.
Yeah.
Okay?
Yeah, Morphozoic. Yeah.
In a sense, that's the general embryology problem.
Yeah.
We get better and better images of embryos in 3 dimensions and over time, but can we invert them into a set of rules by which the embryo builds itself? So the scales are a good place to start because they don't change. The shells don't change in front of you.
Yeah.
Some days ago, I created a video how to create a new sorting algorithm using the mollusk cell pattern. So if you guys want to check, you can check that.
Yeah. Shruti just joined us. We're talking about shells like this.
Oh.
Okay. And the question is, can you go from the pattern on the shell and figure out the rules and make it? It's an inverse problem.
Like that, about this you were talking last week as well, right?
Yeah. So I dug out some of the smaller ones.
Nice, yeah. You can try them out. It's interesting.
Yeah. So we actually, we were talking about Morphozoic, which was the cellular automata approach to it, which is to take like a cellular automata and then take the output and put them into a neural network and classify the outputs.
Yeah.
But that didn't really, get us exactly where we wanted. Mayukh put a link to neural cellular automata in the chat, and that's a paper we went over a while back. This is the Distill publication by Mike 11 et al. on looking at using a neural network or a deep network at the beginning of the process, and it generates patterns that then you put into a CA, and that's the way they approach it. So Debo says, "Can you get the rule from the pattern?" And Mayukh says, "Yes, that's what happens during training." So that's the idea there. And of course, it's not. Maybe it's not exactly what we want, but
the problem with training is it gives you a bunch of neural connections, but they are not the same as rules.
Right.
You don't know what's happening inside there.
Yeah. I mean, how do you interpret? It's like I don't know, translating languages. What rules are you using? Computer can do it by just training it and getting correlations, but I don't think humans do it that way.
Yeah.
Okay, they learn the meaning of the words and.
Yeah. So Mayukh says we can interpret the rules of feature visualization, and there's another. So Mayukh has a thing called differentiable morphogenesis, which I guess is something he worked on in one of his projects. He implemented some neural cellular automata on PyTorch. If you go to I'll share my screen. If you go to the link in the chat, it's right there. So this is his work on it, where he's taking different patterns and turning them into other patterns, which is not quite the same thing as what we're talking about, but this is.
I suppose if you had a constrained set of rules that generated patterns, you could ask for a given pattern which rules can generate.
Right.
That would be a machine learning approach, I guess.
Yeah.
Trying to see which pattern is closest to the pattern generated by rules, and therefore you say, okay, these rules are consistent with this pattern. That might be a way to go.
So he also has this thing called infection models. These models are trained to infect a given CA state. So this is something, Mayukh, that you've done in the last couple months, because it looks like based on the dates that was. So I guess So that's his work. It's in the chat here.
Anyway, there's lots of literature on saying, oh, here's a pretty picture of a snail, and here's a pattern that looks something like it. But there isn't any that quantitatively tries to relate the snail pattern to a set of rules.
So that might be something interesting. I know Shruti actually is preparing a application with some interesting things in that direction. So we can talk about that more. Well, anyways, welcome to the welcome to the meeting. I know a lot of you have been busy this weekend submitting proposals.
Let me make one suggestion. I'll try to hold this steady so anybody who wants can take a picture of it.
Okay.
And see what they can think about. Okay, it's a little hard because I'm using the built-in camera.
Yeah.
Okay. I'm gonna copy that thing.
All right.
Taking.
Okay.
Yeah.
Anybody need more time? Okay.
Well, welcome to the meeting. Yeah. Oh. Was that? Okay. Okay, welcome to the meeting, everyone. I know a lot of you this weekend were working on your proposals, and so those are the deadline, I think, is either today or early tomorrow, my time, which just make sure you check the time if you haven't submitted already, because that's that the time change, the time difference between your location and the time when a proposal is due is often different, and so you want to make sure you have it in on the right hour of the right day. So just make sure that everything, and I sent out some reminders that if you go into the submission portal, for example, and you have a draft, you need to change it to final version before it gets accepted. Little things like that. And so I think if, I've reviewed a lot of people's proposals and they all look pretty good. So good luck on this to everyone. And unfortunately, we won't be able to select everyone, or, we'll have like maybe one or two people at the end who get actually selected for the actual program. But if you don't get selected, I definitely welcome you to continue working on some of this stuff. It's definitely there have been a lot of really good ideas, and we can always turn it into something else. Maybe another, a GSOC project next year, or some other thing. If you are doing work on something that's really relevant to what we're doing, we can pick up on a theme and pull it forward. So there's definitely all sorts of opportunities for continuing with that. So, and then we'll be reviewing the applications in the next couple weeks. And I don't know when the final decisions are due. I think in about a month, and then we'll announce those. And then the community period will start after that. So that'll be, something that we do here. We kind of stress the Open Room Foundation and the things that are going on here. So I think if you've been attending the meetings, you kind of know what the community is all about. So, so I guess I wanted to open the floor to anyone who wanted to present. I know that Debo wants to present some things today. I don't know if anyone else wants to present. If you have anything that you've been wanting to present the last couple weeks but haven't had a chance to, and it doesn't have to be very long. It can be very, just seat of your pants, whatever. So, Debo, why don't you? Are you ready to go or?
Okay. I'm going to share my screen.
Yeah. Go ahead.
Can you see my?
Yes.
Okay, so today I'm going to present a new technique and the approaches to detect and count the diatoms using some RCNN and yellow-based methods, which I get the reference from the counting blood cells. Like there are some papers which demonstrate how you can count white and other platelets, everything using this kind of techniques. So now take a quick look at the project digital basilaria goals. So in this project we are understand the biology of the diatom by collecting the digital data and want to explore more and also we want to create a digital consciousness. So for that we need basic understanding how it works. So for this I think diatom is the most good one, which approach is taken by the development group. So we are going we have take the approach to understand the physiology and neurological process of that basic life form, and also enabling the researchers and other general people to understand the features of the diatom and everything. And the last goal is that to collect the data from the microscopy images to understand the the movement of the of a basilaria colony, and so that way we can understand that and also create some good models or a digital simulation of the basilaria. Now it's a quick some description of the current project state. The project is like currently we have Deep Lab version 3 pre-trained models which extract the phenotypic parameters like describing shape of the cells, and also but they don't have any count method as per I see that. Yeah, and let's go to the next slide. Now, while I am preparing my proposal, so I was searching for new methods to detect the and count the diatoms. So from there, I got this new idea of like some researchers uh, detecting blood cells using faster RCNN, and yellow-based technique is I got that from another, like it's a code base which is open source, and after that like we can took the RCNN based approach to detect white blood cells, and there will be they are using RPN regional uh, potential network to like creating the backbone with a regressor, and after that they will take that like multiple potential region from the cells like this. You can see that detects and send to the proposal, and after that classifier will classify that. And there are some like some experimental result of the like as per the paper, and they also use transfer learning using AlexNet or DTG16, which we also can use for our problem, like just changing the data. And yeah, and after that these are the some photo some images have generated, and this is the link from where I got the code. And but sometimes the model do something wrong, like overlaps or count a same cell twice. So for that they use the k nearest neighbor and intersection over union based verification system, which will find the nearest platelet of a platelet, and after that calculate their overlap, and after that it will eliminate that. Now from that we can adapt these kind of approaches, like detecting the black cell is similar kind of problem to us, like detecting that. Only difference is that we are also want to detect the movement of the diatoms. So we need to some tweaking in the model to improve the detecting and counting the diatoms easily. From here, anyone have any question? Okay, so I think anyone don't have any questions. So now I think about to I and Bradley discuss about the like if we can create a Git book, like a book where we can showcase every documentation, and everyone can easily take a look from there. So yeah, that's the thing, and these are the some references I used, and these are some basic things. So thank you very much.
Yeah. Yeah, that's great. Why don't you leave the slides up? Yeah, so we can look through them. If anyone had any questions about any specific slides or. So I'm interested in your sort of approach, the KNN and intersection over union approach. So this is the thing that. Oh, that's the book. Okay.
Yeah,
that's nice.
Okay, 1 minute time.
Okay. All right. Yeah, I guess he's got to talk to someone for a minute. But yeah, that was an interesting talk. I'm interested in his. Well, that's the book that we talked about putting together a documentation book. We've been trying to find a format for some of this, but that's a Git book, so I don't know. Well, we'll maybe experiment with that and see what it looks like. It looks pretty good though at this point. So Dick had a couple of suggestions in the chat. Well, can you pull your slides back up, Debo? I was going to ask about a couple things. So, Debo, please define your abbreviation. So, yeah, it's important when you present on something to put it the abbreviations, in full. Did you succeed? Meaning, I guess,
like on the data.
were you able to detect the cells with a good degree of accuracy? I didn't really see a accuracy.
I think everything is in the like the open source project. Like yesterday I found this project, so it's a quick. I created the presentation very quickly, so like I don't get the time to make my local system. So that's why.
Yeah.
But I think there will be some IPython notebook which you can use. I will put the link in the comment so you can check.
Okay.
That.
Yeah, and then we're working on this. We're working on a book actually on the mathematical biology of diatoms. So there might be an opportunity to contribute to that. So yeah, and then this is the link to Debo's notebook. So could you pull your slides up again? I wanted to look at some of the couple of the things. So that where you did the thing with the KNNs and that slide 6, I think.
This one. I think.
Well, the next one maybe. Yeah. So that one is okay. So sometimes these models do some wrong predictions.
Yeah, like overlapping.
Yeah, to avoid this, use K nearest neighbor and intersection over union-based verification. So this is a verification system for finding like the overlap between two.
Yeah, like if there will be a 3 or 4 point, then it will just measure the Euclidean distance from its nearest point, and after that it will send you the overlap, like calculating the overlap and try to reduce it.
Okay. So you'd end up with something like a polygon afterwards, or would it be like a constrained circle? Or
Yeah, like I did not make this project. It's an open source code base which I found.
Okay.
So, like after going through the code base, what I understand that is that I try to telling you, but I try to understand it, and after in the next meeting, I try to give a more detailed presentation regarding this.
Okay. And then the slide above it is, what is it? The number 5, I think.
This one?
Yeah, that one. So that's okay. That's where you're doing transfer learning on custom data using AlexNet or VGG16. Okay.
And they are also DMP the approach I found from the research paper. There is no implementation or no code is open source, but they have the regressor with a classifier. So first it will classify the all the cells in blood cells, and after that the regressor will count the numbers of the blood cells.
Okay. Alright, let's see, we have more comments in the chat. So this is contact information for Janice Pappas, who's doing the special issue on the diatoms. So this is some information about her. So if you're interested, this is something you can check into. I mean, we'll probably want to talk about it some more, but just to give an idea of what how you might move this stuff forward, and yeah.
But it's this page is missing.
Oh well, we could. I have her contact information too, so we can talk about it later. But Mayukh said, as far as I remember, the classifier determines which classes are present in their image, and the regressor estimates their positions, width, and height. So the classifier actually determines the sort of what category they belong to.
Yeah, like the platelets or the white blood cells, like the types. of the
Okay.
blood cells. Okay.
So they're doing both at the same time. They're estimating, like, yeah. Okay. Yeah, that looks good. Thanks for presenting on that.
Yeah.
Yeah.
First time I'm presenting something.
Yeah. No
And
problem.
Bradley, have you take a look at the draft I send you, like the kit book one?
Yeah, I looked it over. It looks good, but I have to evaluate it a little bit more. But it looks pretty good.
Yeah.
I know it's a continual quest to try to find like ways to display like documentation and publications and books and things like that online, because this looks like a good solution. Though we'll have to work with it a little bit more.
And also, like, if you want, like, it has the kit book has, like, you can invite other people so they can also collaborate with you, and as well as I think there will be something similar to version control like it.
Yeah, I would imagine.
Yeah.
It should also display equations in LaTeX to some degree, right? I mean, because it's, well, is it based on GitHub or is it like its own platform?
Yeah, maybe, I think.
I mean,
later I will check that, if that is any support for the LaTeX.
Yeah, I mean, yeah, these are, but this is sort of like the read the doc structure where you have the menu on the left and yeah. Yeah. Oh, that looks pretty decent. Yeah.
Yeah, like there. I just copy and paste the documentation because I don't have much time, so that's why I just create this basic draft. But later on, I try to create a dedicated tutorial for like how to run the project.
Yeah. Yeah. Well, yeah, I think. Oh, yeah, and that's the README that we have.
Yeah,
so
that's the
there. Yeah. All right. Well, very good. Thank you.
Yeah.
So does anyone else have anything they want to present today? I don't know if Mainak, a couple weeks ago, he said he had something he might present, but I don't know if he's still here. Let's see. Okay. So Ayush says, yeah, the regressor tries to learn the numeric values for the bounding boxes, and the VVG backbone tries to classify the object inside the bounding box. Jesse says, Git book looks cool. Ujjwal says, regressor can be used to classify only when classes are continuous, which is not the case here, I believe. So there's a continuous versus discrete issue, with... Yeah.
Yeah, maybe, yeah. That's the problem.
Yeah.
That's a downside, but I think we can figure out something like counting because the movement or the diatom colony has a number. Like that's a discrete property of the diatom colony. So from that we can figure out some equation or something like that, like some model if there exists, we can use that. So that's why I told that we can tweak the model as per our need.
Okay. All right. Yeah, I mean, there are different ways you can do it. I know people have come up with different tricks, and sometimes they're in the different field statistics literature. Like, the psychologists and economists come up with all sorts of interesting ways around the sort of the assumptions of these statistical models because they have different types of data from, what a statistician might assume is the, the perfect type of data. So that's something, that they... But I'm sure that, there are ways around it. It's just a matter of recognizing your limitations of your data. So... So Mayukh says, sadly, Gitbook does not support themes unless we use the paid version. A great free alternative is Hugo. So Gitbook actually has a paywall on it at some point. Read the docs also be a good alternative. Yeah, read the docs, of course, is basically very similar. And Mayukh put a link in the chat for Hugo. So this is where you want, if you want to use themes, and there are different themes for that.
Yeah,
So I mean, yeah,
lots of things.
yeah. So let's see. Vrutik is here, and love Vrutik. Ujjwal's here. Ayush joined us. Jesse's been here. Mayukh. Krishna's here. Vash is here. Yeah, so let's see. Oh, okay, Mayukh says they support direct markdown. Yeah, I think if we just create, if we have some good content in markdown, we can put it on whatever platform we want. So I guess we'll then move on to some other business if no one else wants to present. I'm going to present my screen again. Oops. And not open my chat again. What was that?
I'm Vrutik.
Hi.
I'm a GSOC applicant.
Yes.
And I've been working on central extraction from CL1's images.
Okay.
So I want to present it, but I had some issues. So is it fine if I present from my phone? Right?
What do you mean? Oh, yeah, you can do that. Okay. Yeah, let's make it read the docs after GSOC. Yeah, we can. So DevoLearn read the docs. OpenWorm has a read the docs, and I have some information on DevoWorm on there, and it's a little dated now, of course. But yeah, we should make one for DevoLearn because we've not that needs to be picked up again. So Devo says that's a great idea. Then Dick put a book citation in the chat here. This is about diatom identification double challenge called ADIAC, and that's this in the Proceedings of Image Analysis and Processing. So this is from 1999. This is this like a challenge, call to action, one of these grand challenge type things, Dick?
I think it's a summary of the book about ADIAC, and I can't find the book offhand.
Oh, okay.
And one question, OpenWorm is signed up for the GSOD.
Yeah.
So there we can give the project to create the DevoLearn.readdocs.io project.
Well, yeah, GSOD, I don't know. That's a little bit different this year. So the projects are already submitted, and they're not I don't know how they're doing it this year. It's like they've got a group of people they already have selected, and they're doing I didn't put in this project. I didn't think of it at the time. So, that's something that we probably wouldn't be able to do much with this year, but
next year we can do that.
Maybe,
but
yeah.
For now we are going to do our own. I think that will be great.
Yeah, I think that's great. Yeah. So, Vrutik, are you interested in presenting, or did you say you were going to do it another time? Okay. Oh,
Actually, I'm searching for a present option on my phone, but I'm not getting it. So I think I'll share the slides link, and I'll just say what I'm doing. So
okay.
I'll just say the sort of thing that I'm getting the slides link.
Okay.
So hello everyone. What I have been working to extract centroids from C. elegans images. I have pasted the slides link in the chat box, so it would be nice if you view it. So what I have done is we have different machine learning models and all to segment and extract the centroids, but I have used a slightly different approach because we had images where centroids, means there were nucleuses overlapping each other. So there are some algorithms failed, some models failed. So what I did was I took the images, I locally enhanced images. Like if we have a pixel, I added the densities of surrounding pixels, so which would eventually help me to get the center pixel of a cell to the highest density. So that's what I did by taking an candidate region and taking some few diameter dimensions which would be possible. Like we have maximum 80 pixels, we had maximum 80 pixels of dimension in our data. So I took like an interval in intervals of 4, as many as dimensions as I could take, and I used a sliding window technique to get the locally enhanced images. The images you can see on slide. The image I am talking about
Slide which one?
Is on seventh slide number, 7th and 8th. You can see the difference. Like we have the center pixel at the maximum intensity, and then what I did is like I used a sliding window technique where I compared pixel intensity with the surrounding pixel intensity and chose the maximum intensity and took that point as a centroid. The problem I faced there was like when I suppress the noise doesn't get completely suppressed. So I kind of like you get more number of centers, redundant centers which you don't want. They are generated due to noise. Like if we have a single pixel and all.
So total cost, are you ready for this?
I think Vrutik dropped off for a minute here. Well, he's got a lot of slides here. If you're looking through them, put the slides in the chat. And he's talking about refining the centroid, so he's done a lot of work on that. He's got output, which so this says no training or data set annotation required. Gives accurate results by manipulating pixels of the image, and it works on the images of embryos of other species with minimal or no change. So this looks like an interesting algorithm. I'm not sure. I don't know what happened to Vrutik, but it looks really interesting. I just hope he's able to join us again. I don't know what happened here. Let's see if he's on. He's on, but I don't know. He might have dropped off and he wants to come back online here. So yeah, this is very good work, Vrutik. Yeah, so look through these slides if you're. This is, I guess, one of the papers that he was going through to work on this. Future developmental possibility. The current algorithm is implemented on 2D images, due to which there is a loss in data due to varying pixel intensity, which arises due to different z-coordinates. So I think maybe it was Vrutik we talked about the z-coordinates, or maybe Yash as well. That you have this stack of images. So when we're using the raw images, there is a z dimension, but it's hard to infer.
So I got just disconnected.
Oh, okay. All right. So yeah, why don't you finish up your Why don't you finish up your slides? I was just talking about them a little bit.
What I was talking about at the end, please, can you just tell me?
I think you were talking about this, like manipulating the pixels and, yeah, I think you were at this slide here, number 8.
Yes, exactly.
Okay.
Yeah, so what we have is like, this is the original image and we need a locally enhanced image. So what I did, you can see the square. and I want the center dot to be at maximum intensity. So I would take a cat region and I'll select the images and I'll add all intensities around the center to the center pixel. So what it will generate, you can go to the next slide, please.
Next slide.
Hello.
Yeah.
Yeah. So yeah, this is image where like the intent middle pixel as high as possible, so that we can use it further. Now on this image, what we would do is viewing window, and we would we would take the pixel with the maximum intensity as a center. So what we do in this image, it seems that all but it's not of go to the next, see the output of please can you change, yeah. So here you can see like there are many centroids which we don't want actually. These centroids are generated due to the noise, as we have suppressed it due to Gaussian with Gaussian and median filter, but it does not get suppressed completely. There is some noise due to which these centroids are generated. So to discard this centroid, keep what useful one. This like map to low and I'll have a threshold value. Like if the threshold value of means sum of intensities is above this, then only it will be considered as a center. So if you go to the next slide, you will be able to see that. So yeah, these are side by side. So I have mapped each and every centers to the right image, which is locally enhanced image. And please, can you switch to the next slide? Yeah, so we got these centers. These are the useful centers, but the problem arises here is there are means few centers detected really close to each other. Like for a single cell, there would be multiple centers detected. So we have to keep a minimum threshold to filter. Like if we of 5, so what I did like distance Euclidean all the sets, and I showed I means discard the distance less than or equal to 5 pixels. So when you go to the next slide, I have mapped each and every center detected on the image. Yeah. So we got this output. Now, as this is a 2D image, there are no Z planes involved in it. So you can see there would be some like if two cells are overlapping, you will get converted into locally enhanced image. But if the cells are not overlapped, you can see there are 4 cells nearby each other. Still, we are getting 3 of them, and one is missed due to the smaller cell is overlapping the larger cell. So we can we have to means consider 5 to 10 pixels. Here it may be large because of the overlapping cell, but when I did the same thing in 3D by taking the Z coordinate also on the images obtained from EPIC dataset 2, which had 31 slices in Z coordinate. So it worked really fine, like it gave me output with errors of like 10 to 15 plus minus 10 to 15 of pixels, and in Z coordinate it was always a whole number because I went slice wise. So this was what I was doing, and I have been optimizing this algorithm on 2D images. It works, it gives the output in 0.14 seconds on GPU obviously, and I'm just improving it further, the space complexity and time complexity of the algorithm. That's the same thing has been implemented on 3D images also. I'll be sharing some collab links if you want to refer them. Thank you.
That's very good. Thank you. Thank you,
So,
Vrutik. Yeah.
Yeah, these are the with the outputs. This is the article which I referred. This was the reference of my algorithm. They did on like mouse embryo. I tried to implement on C. elegans.
Right. So you could do this on pretty much any cell data set where you're trying to find a cell in a background. Yeah.
Yes.
Yeah, I think, thank you for that. That's good. I think, Vrutik and Debo have some things in common there in terms of like looking at like how you deal with noise and overlap. And that's an important issue. I mean, it's easy to think, well, you can just, follow the boundaries between cells. And sometimes, in some of these data sets, you're just taking a slice of the sample. And so in doing so, you're going to pick up, overlapping cells and things like that. And especially when it's not something, even with an embryo, you have that, but especially if you have like, some group of cells that are in a colony or maybe, in different layers that are kind of interdigitated, you're going to have to deal with that issue. So I think those are two very good solutions to that. If you could share your notebooks or whatever you have, and maybe put them in the Slack as well, I think people would appreciate it.
Sure, I'll be sending them on Slack channel.
Okay. Thank you. And then Dick put some more references on
Thank you again for presenting my slides and helping me.
Oh, yeah, no problem. Dick put some references to IDIAC, IDIAC. So this is this approach, and so there's a lot of stuff on diatom identification here. Different techniques like binary masks of concentric rings. So people have been doing this for a long time, coming up with different techniques for, segmenting images and things like that. So you might take a look at some of those references. There's a lot of background noise somewhere. Okay, thank you. So yeah, check that out if you're. Well, I would check it out. Yeah.
Yeah, I'd like to present something.
Okay. Is that Krishna?
Hi.
Yeah. Hi.
Yeah.
Okay, go ahead.
There is some background noise, I guess.
Yeah.
Someone put some
Okay, thank you. Yeah.
Is my screen visible?
Yes, it is.
Yeah. Hey, Krishna.
Okay. This is nice.
It's just a small thing. Would take more than 5 minutes.
Okay.
So it was the paper you, me, and Jesse are working about.
Oh, yeah.
So I did a small portion on it, and it is known as correlation doesn't imply causation. So if we have two variables or two events that are having some correlation between them, it doesn't mean that they are having some causation between them. Like, so one example from Wikipedia is sleeping with one shoes is strongly correlated with waking up with a headache. So therefore sleeping with one shoes cause headache, but that's not true, because if a person is like sleeping with his shoes on, there are chances that he can be drunk or he didn't have a good sleep. That doesn't have anything to do with the shoes on. And this is my favorite example, one of my favorite example, that like when the sales of soft drinks increases, therefore the deaths of drowning also increases. Therefore soft drinks cause drowning, but that's not true. You can say the underlying reason between these two correlation is that soft drinks are usually sold in summer temperatures, and in summers people usually tend to go to near water, go for water sports and go for swimming. So it's going it's the temperature that is causing the drowning, not soft drinks. So am I being audible?
Yes.
Yeah. This is an example. It shows that global average temperature and number of pirates. So if one can see this, it can be easily shown that rising temperature is kind of wiping out pirates, but that's not true. It's the advancement of the civilization. So it has nothing to do with the global warming. It has to do with the how the civilization is progressing. So one can these charts maybe depicting some relationships, but it's a third variable that these charts are more connected to. Like pirates and global temperatures don't have any correlation. It's the advancement of technology, it's the industries, it's the global trade that corresponds to increasing temperature and we can say falling of pirates. So, for example, there are so many things that usually show correlation, but they are not a causation. Like I was seeing that when the in a city named Kentucky, when the sale of boats, fishing boat increases, there were also more marriages. So one can deduce that okay, selling of boats and buying of boat is causing marriages, but it's not true. It's actually the marriage season and the fishing season is overlapping. So it is the third variable that is causing the causation, not the relationship between two. So like marriage and selling of boats is one can say that independent from each other. So is the temperature and the pirate, but they show some correlation and it can lead to some bias and it can lead to bad decisions, but that should not happen. So we can have possible solution that is intervening some human interventions. So we can we can justify that the correlation is just you can say happening randomly, or it has some story behind it, but it's not the causation. That's it.
Okay. Yeah. Yeah. There's a lot of literature on that. You can even get lost in it in terms of like what's a neat correlation you can make, or what requires a larger scale analysis. Like if you're looking at the intervention of different variables, or maybe you have like something that's just like correlated because they're by chance. Thanks for that. That's good. So that paper, we're working on. It's actually more in our other group, but we're we'll talk about more about that later. But I think that was useful for this group. I wanted to talk about some things. Shift to something. Actually, let me go through the little chat here. Another example: all deaths are due to COVID. Therefore, we are otherwise immortal. Ujjwal says, how about all deaths because of COVID? Premise is not correct, I guess, in this case. Dick says, because anyone who dies is now attributed to COVID by politicians. Thank you for the update. Krishna gives a thumbs up. So, So, I wanted to move on to a couple other things here. I wanted to mention that Dick and someone who works in the other group, Rob Stone, they have a new paper. And I wanted to show that paper real quick. Let's see if I can find it. So this is a paper in the Biosystems Journal. It's in one of the special issues. It's in this differentiation waves special issue. And this is on something called Janus phase logic. So this is a short tutorial on the Janus phase logic of differentiation waves and differentiation trees and their evolution. And so differentiation waves in trees, we talk about it somewhat in the group, but a lot of you probably don't know much about what they are. Dick has a huge book on this topic from 1999. And so, but instead of going through the whole book, you can read this tutorial and it gives you some idea of what those phenomena are. And so, the abstract says differentiation waves offer a different perspective on causality and embryogenesis from that of molecular developmental biology. So, going back to Krishna's thing about causation and correlation, we also deal with causality, and of course, causality is a minefield in and of itself. But in this case, we look at like the embryo and we have a lot of the genetic networks mapped out and things like that, but we still don't understand maybe the causality at the level of the cell. And so this gives us a framework to think about that. And so Rob, I think this sounds like Rob's phrasing here. Janus face cybernetic logic, which Janus face is I think it's from Roman mythology, where you have two faces, and it's like good and bad or good and evil or something like that. And so it's it's a binary opposition. So I have some things in the chat. Oh, thank you, Debo, for attending. Dick put a link to his book in the 1,760 pages. He also has a shorter book here, but this, of course, and Jesse says looking ahead and behind. So that's the Janus face analogy. But the important thing here is it's a binary opposition. So with local and global top-down, bottom-up dynamics, this is the Janus face approach. It's not reductionist. It's distinct from emergence and outlines the process theoretically. So this paper goes through a lot of this stuff, kind of describes it, and if you're not familiar with some of these concepts, it gives a little bit of background into it. Although you'll still have to look things up. I encourage, if you read the paper, to look things up. And they walk through a typical, what we call a differentiation tree, which is where you start with a zygote and you get these differentiation events where you get the differentiation of tissues, sometimes of cells that divide and then differentiate into different functional lineages. And that's what this process here. And these coins are the Janus faces. So at every step in this process, you get this binary faced decision. So you have a switch that happens here. And so, I'm not going to go deeply into the paper, but I'm just going to say that this is it's pretty digestible. We have a number of consequences for evolution here, which is talking about, different things in development that this model of development has a consequence on. So, for example, there are things like heterochrony, or the evolution of neoteny, or metamorphosis, which is when you have one organism that transforms from one phenotype to another in development, like a caterpillar into a butterfly. You get different types of asymmetric cell divisions, and so there are all these different things that it explains. So I think that's I'm going to put the link to the drive here again with the papers in it, and I'll put that into the chat. That contains this paper. Thank you, Ujjwal, for attending. Yes, Ujjwal says, make sure you submit your final proposals before the deadline. And once again, a reminder. And then Dick says that those gold little circles were Roman coins with the Janus face on them. So they used to have coins with the Janus face on them. I wanted to go to some other things. I have a couple other things to present on. I wanted to get into, or kind of highlight them. Susan sent me this link to this talk by Michael Levin, who is a very prolific developmental biologist, computational biologist. And this is a TED Talk he gave back, I don't know how long ago. I think it's fairly recent, but this is on a lot of his work on looking at bioelectricity. And so Michael Levin works on these model organisms called flatworms, and you can see them here. They have their... And the thing about the flatworms is that you can take a flatworm and you can cut it in two, and it doesn't not only not die, but it forms two different organisms. So they're basically they're clones of one another. Not only that, but you can take everything but a single... You can obliterate everything but a single cell, and then that single cell will regenerate the worm. So it'll regenerate the entire flatworm. Not only that, but you can take a single cell from the flatworm you can train the flatworm, then you can obliterate everything but a single cell, and that single cell will it will regenerate back into that flatworm that actually can retain the memories of the flatworm, the original flatworm. So this is really a fascinating model system. And he studies bioelectricity, so he studies the bioelectric communication between cells, and this is basically very similar to what neurons do when they have an action potential. So action potentials are a type of bioelectric communication, but they're very distinct signatures that come from these neuronal cells, but you also observe electrical activity in non-neuronal cells, like in skin cells or in liver cells or any other kind of cell you can think of. They have some electrical potential, but it's not like a action potential where you have a spike. It's just some sort of electrical signal, and so that electrical signal, you can study that and see if it has an effect. And he's finding that it does have some organizing effect on the phenotype. So, if you have a bunch of developmental cells that differentiate and they have this electric potential, even stem cells have an electric potential. That electric potential is used to coordinate the cells and to form features of the phenotype. So he does a lot of work with that, and it's a really interesting area. And he talks about this in this talk. But he also talks about this thing called xenobots, which are this new model system that he's developed with some collaborators. And this is a type of single cell. It's almost like a biological robot that they've developed. I don't think I have a paper on it this week, but we'll talk about that in a subsequent week. But anyways, this is really fascinating. Thank you, Susan, for forwarding this. You should check this out. Another thing that I wanted to talk about was this article. So we've talked about cellular automata, and we've talked about like how we might be able to use them to find, discover how pattern formation works and things like that. In this article, this is an interesting article I found on the Medium. I think it's just a Medium article, but it's on making stem cells using AI. So it's kind of an interesting foray into sort of cellular regeneration and then building computational models of things like regeneration, and then kind of going through some, like they show a lot of examples of regeneration in say like amphibians and salamanders where the tail gets cut off and it regenerates a tail. And, you're trying to boil this down to like a set of principles. And so this is something called autonomy of cells. So cells are, they have to behave locally and fill in the gaps, so they regenerate this tail. And then it gets into cellular automaton and talking about like how cellular automata can regenerate. So you can change from a 7 to a 2 given some differential activity on the grid of the cellular automata. Then it kind of goes through. So this is like a tutorial, but it kind of brings up some really interesting issues. Talks about neural cellular automata. So you have this combination of the cellular automata grid and neural networks. And so this is a good tutorial. I recommend, like, if you're interested in this area, to read through this. Discusses a lot of the issues. And I don't know if it links to a lot of these the still articles that we've been following, but conscious MNIST digits, which is kind of interesting. That uses the word conscious, but I'm not a big fan of like using the word consciousness when you mean like something that's cognitive. But anyways, so this kind of goes through a lot of this. Why is this useful? So this is like self-organizing MNIST digits. And then here's some resources which are interesting. So here's a link to that. And let's see what we have in the chat here. Dick says, but what deep knowledge does a flatworm learn? Well, they don't really learn any deep knowledge. I think they know like basic like associative memory and things like that. It's not, but you can tell because they learn associations. I'm not really sure. I haven't read the paper in years, so. the electrical signal is often due to calcium channels on the surface of cells. Susan, yeah. So Susan, yeah, they generate a number of potentials in the cells. Dick says Lionel Jaffe found huge currents in chick neural plate, which may correlate with the differentiation wave for the neural plate unexplored area. Susan, salamander tails do not always grow back exactly the same way when they regenerate. That's true, and there are a lot of environmental cues, and we talked about environment a couple weeks ago. What does it mean? And one of the things I guess it means is that it's an organizing signal. That's still pretty vague, but I think that's something to talk about too in more detail, because that's true. They don't grow back the same way. They just make do with what they have. The cues that they have. And then Krishna says, my problems are like flatworm. I try to destroy them. They multiply instead. Okay. You have regenerative problems. All right. So those are two. I wanted to bring those articles up. I also wanted to point out that we have a new acceptance, and we have our submissions file. I talked about the different submissions and opportunities for submissions. One big update on this is that Networks 2021, this abstract has been accepted. So this is going to be a presentation at Networks 2021. This is on embryo networks plus connectomes, and we can talk more about this later as this is being developed. If you're interested in collaborating on this Networks abstract, and this is I think the abstract link here, let me know and we can work on the presentation. You can get mentioned in the presentation. So so I think that's it for that. I just want to mention that. And then if we go back to the papers, I wanted to talk a little bit about a couple more one or two more papers before we go today. I think this is thematically correct here. So the first one is this learning and synaptic plasticity in 3D bioengineered neural tissues, and this is by a number of people, including, again, Michael Levin. And so the abstract on this paper reads, though neuroscientists have historically relied upon measurements of established nervous systems, contemporary advances in bioengineering have made it possible to design and build artificial neural tissues with which to investigate normative and disease states. However, their potential to display features of learning and memory remain unexplored. So here they demonstrate response patterns characteristic of habituation, a form of non-associative learning, in 3D bioengineered neural tissues, exposed to repetitive injections of current, which is electrical current, to elicit evoked potentials, which are the types of things that they're not really, they don't have to be bona fide action potentials, but they're things that show up and you can record and at the regular electrical responses of the cells. A return of the evoked response following rest indicates learning was transient and partially reversible. Applied pattern current as masked or distributed pulse trains induced differential expression of immediate early genes. So what they did was they took current and they delivered it in different patterns. So they either deliver it in a bunch of trials almost simultaneously, or they distribute it over time as a series of pulses, like maybe once every 2 seconds. And then they're able to do this and look at the effects. And in both cases, they induce differential expression of immediate early genes, which actually are like an early marker of synaptic plasticity. They're genes that respond to stress. It's like the first set of genes that respond to stress, and people have studied immediate early genes in a lot of different areas of gene expression, but also it's a signature of plasticity and memory formation. And so this is the first demonstration of learning response in a bioengineered neural tissue in vitro. So this is not the flatworm context, but this is actually something much less organized. This is a synthetic neural tissue they've engineered in the lab, and they're able to actually just kind of elicit some response from that neural tissue by delivering an electrical stimulus. And in another Slack organization, last night we had a discussion about the Neuralink brain-machine interface, the one where they're just delivering information to the organism via like a remote control or like a wireless setup, and then the organism is behaving. And so we had a discussion about the role of maybe something like embodiment, or the role of like what the stimulus input should take. Is it just okay to deliver or is it just enough to deliver electrical potentials or some sort of signal to the cells, or do you need the entire body, whether it be a flatworm body or a human body or a primate body to facilitate the proper response to that? Well, in this case, they're not really looking at behavioral outputs, they're looking at electrical outputs. So I don't know, I just brought that up as an aside to tell you that this is a very simple and contrived model system. So they kind of walk through this protocol, they walk through how they record the electrical activity, then they show this example of how they build this culture, this bioengineered cortical tissue. It's a ring, and you get these different silk fibers that serve as a scaffolding in a hydrogel. And then these, they have neurons that sort of build connections along those scaffolds, and then they're able to apply a current and actually do these experiments. So again, this is a very contrived model system, and they walk through a lot of the results. They show the masked and distributed patterns of stimulus input. So this is where you just deliver 60 pulses all kind of in a rapid sequence. And in this case, you have a distributed pattern where you deliver 20 pulses and then wait a minute, and then 20 pulses, you wait a minute, and then 20 more pulses. So you have this, this refractory period where the network can forget about these pulses to some extent and recover from it, and then they can maybe build a memory of what these pulses were like, and then experience the next set of pulses with that memory of these pulses. And there's maybe some anticipatory response, whereas in a masked pattern delivery of the stimulus, there's no chance for the system to build any sort of anticipatory response, and so it's just kind of being hit constantly with this pulsed electrical activity. So you would expect to see differences in maybe some sort of non-associative learning. So these are the references. So this is in this folder that I shared with you earlier. And then finally, I'm going to talk about this paper here. Again, this is just a thematic thing on my part. This is where they use these sea slugs called Alicea. This is a model organism they use in learning and memory a lot. And they basically, what they did here was they induced memories by taking the RNA out of a trained Alicea, and they put it into a non-trained Alicea, and the non-trained Alicea or sea slug was able to learn things through that transfer of RNA that they wouldn't have learned. Or actually, they're doing it in a plate. They're doing it in a dish as well here. So they're taking, they're doing it in a sea slug, and they're doing it in culture. And so they're able to show that the RNA is responsible for transferring the memory instead of something like learning, or experiential learning, where they're interacting with their environment. So the significance statement in this paper, they have a significance statement. It is generally accepted that long-term memory is encoded as alterations in synaptic strength, and this is what we use in our neural networks as sort of a rule of faith, an article of faith. An alternative model, however, proposes that long-term memory is encoded by epigenetic changes. So what they call non-coding RNAs can mediate epigenetic modifications. Therefore, RNA from a trained animal might be capable of producing learning-like behavioral change in an untrained animal. And so RNA is just basically these molecules of expressed genes. They come from genes that are expressed and they're turned into RNA. And then you have this concentration of RNA in some sort of buffer that they can transfer to another organisms' cells. So it's a very, they're basically, this is the thing that's encoding, this is the thing that they're looking at. Instead of looking at synaptic strength, this is the influence. And so, so they're saying that this may produce learning-like behavioral change in an untrained animal. So here it is demonstrated that the memory for long-term sensitization, which is the model that they use in Aplasia, this mollusk, and it's just like it's like, I think, a gill sensitization where they learn different patterns of opening and closing of the valve, or the, so it's not like this this huge set of thoughts or this consciousness. It's just this very simple behavioral adaptation that they have. And so, but nevertheless, it can be successfully transferred by injecting RNA from sensitized into naive animals. Moreover, a specific cellular alteration that underlies sensitization in aplasia, sensory neuron hyperexcitability, can be reproduced by exposing sensory neurons in vitro. So that was that dish that they showed in the figure, to RNA from trained animals. The results provide support for a non-synaptic epigenetic model of memory storage in aplasia. So this is, again, is a model organism for learning and memory. Aplasia californica, which is a it's a common model organism. They have, they basically train this they train one of them in this, habituate it basically, so they expose it to some stimulus and they, observe this habituation. And then they're able to take, they test the control animal first and they test the trained animal, then they make the transfer of RNA to the control, or the naive animal, and it exhibits the same behavior without actually experiencing this training firsthand. And so they can become habituated without the experience. But then you can also do this in a culture with neurons that are disembodied, and you get a similar result. So I think this is a really interesting paper. Again, they do a lot of electrophysiology, which, that's going to be a barrier to understanding exactly what they did, but I think you can get the idea. And so they show some results here and the references. So that's going to be in that folder as well. So now I have a bunch of things in the chat here, but we'll go through them before we wrap up. So Dick has the Jaffe and Stern citation there. Strong electrical currents leave the primitive streak of chick embryos. So that's a science paper from 1979. Vrutik says, I got a really annoying network issue today. I need to leave. Well, thank you for attending, Vrutik, and thank you for your presentation. Catch up on YouTube. Thank you. See you next week. Yash also had to leave, and thank you for your presentations, Vrutik and Debo. Jesse says, yeah, I think there's a point to be made somewhere about the sort of embodied radical, embodied cognition stuff. Is something being used as an analog or a transference, or is it doing anything beyond that? Moving with the cursor with your mind just being remapped from other movements. Which is, it's an interesting question. I just showed you two papers where they're sort of addressing the idea of, like, how can you have this thing called learning and memory, which is actually something that maybe is a broader class of things, behaviors in cells, maybe electrophysiological behaviors. And so we don't really, we assume we know what learning and memory is. It's and then we say, well, it's learning and memory. Well, I'm showing you an example from cell culture where they're doing basic sort of maybe some non-associative learning things, and maybe from this marine organism where they're doing this habituation behavior. Okay, thank you, Tharun, for attending. But then, that's not really the same thing as, like, maybe learning how to control something with your mind. In the Neuralink work where they have this, I think it's a primate, or it could be a rat, to control something with your mind at that, in, say, a mammalian brain. And so that's quite a different thing. But nevertheless, I think we can learn things about that. It's really interesting how, we kind of think we know what memory is and we know the role of experience, but do we really know that? So, again, that's something, yeah, Dick says you are who you eat. And which is interesting because I think they've done other experiments where they've looked at, like, different things that or smaller organisms if consumed in the environment. In fact, I think it was flatworms, where they consume things in the environment. They can actually learn from that because they're consuming, I guess, the RNAs of other things, and I don't really know, but there is that. I can't remember what paper that was, but I remember reading a paper about that one time. And again, this isn't like really complex learning. This is like a very simple form of learning, but nevertheless, Worm Runner's Digest. Okay. Yeah. So this was the theme of Worm Runner's Guide in the 1960s. Okay. Well, thank you. Yeah, that was a good one. I have to look that up, actually. I don't really remember that. But, yeah, I mean, I wasn't alive then, so I don't know. But, I mean... so, yeah, thank you for attending. Anyone else have anything they want to add before we go?
You're not that old, are you?
Yeah. Okay.
Yeah. The Worm Runner's Digest is half jokes and half real science, and you couldn't tell which half was which.
Oh, yeah. Open-ended satire, yes. All right. Well, thanks, everyone, for attending. And next week, we'll see if anyone has anything to present next week. You'd be welcome to present.
I think it went the way of the Journal of Irreproducible Results.
Oh, yeah, yeah. All right. Have a good week, everyone. Bye.
Bye, Susan. Bye, JP.
Bye.
Great. Join YouTube.